so is Pappagallo Noemi.
Yes. Fine. So I am Noemi Pappagallo. I'm here to present my research project for the priority theme, the first priority theme about the Kemat Sustain project. There is referred to the methods, to the creation of new methods for safety and sustainability assessment of chemicals and nanomaterials in the European Union. I have divided my presentation into four main topics, but I don't list them now because they will be considered in a moment in detail. So I start with the project partners. This is a European project, so it involves different universities and institutes around the Europe, and of course the University of Urbino is among them, with the project coordinators that are from the University of Hamburg in Germany. What is Kemat Sustain project? Well, nowadays we have a lot of data about materials, but when we refer to material at nano levels, their property, their effect on the environment or on the human health can change. So can change because the size or the shape changes. So there is a lack of validated in vitro tests for a variety of materials, and this leads to a lack in data about their safety and sustainability. So Kemat Sustain has been developed, was born as a project to identify, to test, and to deploy a new innovative method to improve, on one hand, screening and testing protocols, and on the other hand, risk and sustainability assessment in the European Union for a variety of materials. These materials are chemicals, that is the C in light blue here in the name, and nano materials, that is the M here in the name. This is like a brief slide in which I wrote like a little bit of the context in which the Kemat Sustain project was born. So the European Union signed a Green Deal and published the chemical strategy of sustainability that gets operationalized by the Safe and Sustainable By Design framework. This is a summary slide in which you can see like flowchart, and this is how the project is organized. Everything starts with the selection of the chemicals and nanomaterials, so the C and M's, I will refer to them from now on as C and M's, followed by the preparation of them, phytochemical characterization, biological response, and so on. And all of this data will be put into the database and will be analyzed thanks to the AI, so the artificial intelligence that will match all of this data and will like make a prediction about the CNMs in order to create like a footprint for each of them. And in the end, there will be various actions that will be done, but I want to focus on just two of them. That is the creation of the standardization and validation dossier. That will help the European Union to make strategies and to regulate these kind of materials, and the creation of the Kemat facility. Why? Because as you can see, we have a lot of like research outcomes, because there's partner with a chemical background, with a biological background, with an expertise in sustainability, and so on. So the key role of the facility is to transfer all of this knowledge, not only among academia, but only like among private sector, so industries and factories that produce and use these CNMs. These again are the research aims I've always told you about, and in a nutshell, I have like highlighted the primary aim of the product, that again is to standardize screening and testing methods for CNMs in the European Union, and the last one, that is the dissemination of the results. That is mandatory. So in the future, we will share this like results with the external environment. The University of Urbino is in charge of various activities, so a lot of activities will be tackled. And for example, the collection of the photoelectron spectra with the UPS technology, again, the artificial intelligence, but I want to put your attention on the last highlighted and underlined sentence, that is the evaluation of metabolic response of human cell exposed to CNNs thanks to the microfluidic system. Well, the project has pre-selected a huge list of materials to test. And among them, there are molecules, thanks to which the logo has these green parts that are molecules with a plant origin, triterpenic acids and aromatic acids. The last one will be purchased rather than the triterpenic acids that will be obtained thanks to a patented method that uses the pulp, the pulp that its derivation is from a variety of pulp called red sentinel. This method, the patent is down here. I don't see. Okay, down here, and this technique allows the production of a large quantity of triterpenic acids that doesn't vary with the seasonality. And this is very important because what does it mean? What does it mean? It means that the production of acids still remains constant and stable during the time. And starting with the pulp of the fruit, it's created a cell culture called callus, and this callus is composed literally of plant stem cells. This is the quantitative analysis. These are all the triterpenic acids, and in particular the aromatic acids. The quantity of the aromatic acids is 400 times higher than the quantity that we can obtain starting from the fruit on its own, not from callus or cell culture. Of course, we know from literature that triterpenic acids have a lot of biological activity, anti-inflammatory activity, anti-diabetic, anti-hypoglycemic, and so on. And all of these features of them are useful, especially in the nutraceutical, pharmaceutical fields, to create supplements, products, and so on. Here I attached a part of the protocol for of the extraction method from the callus pulp of the red sentinel that is easily shared with the project partners thanks to a QR code that is here on the right. This QR code is printed on the label, and the labels are attached on each sample. And why this is important because the extract will be shipped to partners in order to allow the test and the analysis. And where will this extract will be tested? They will be tested in the microfluidic system that allows a refinement of the in vitro model because mimic the in vivo environment. Why? There is a mechanical stimulus imposed by a peristaltic pump, and the medium flows into these pipes inside and reaches these blue chambers in which we have the cell culture. So this this movement, it's to mimic the blood flow, and inside these chambers, the EAHY926 cells will be used. These cells are somatic cells that can better simulate the condition that we can find inside these because of their derivation of the human vascular endothelium. So mimic the vascular conditions, so they grow adherently, not in suspension. And the analysis that will be performed, of course, the cell viability, so the toxicity of a substance with the Triton Blue, and the analysis is automatized thanks to this kind of instrument, and the evaluation of the inflammatory markers with the QRT-PCR. the inflammatory or anti-inflammatory activity of the compounds tested on cells will be evaluated, taking into consideration the microRNA 146A that involves the toll-like receptor 4. Well, I'll be brief in this case, but the toll-like receptor, the activation of the receptors, makes possible the creation of a complex that involves the interleukin-1 receptor-associated kinase 1 and the TNF receptor-associated picturesque adapter molecules. And this kind of complex stimulates cascade signaling, and in the end, the NFKB transcriptor factor is activated that encode that induce the expression of genes encoding pro-inflammatory mediators like the interleukin-6 and the microRNA 146A that works with the negative feedback on its targets. The molecules circled here will be the molecules that will be evaluated by the QRT PCR. And in the end, I have imagined the research over a 3-year period organized like this. So the first year, the culture of how this is preparation of the extract, the shipping to partners, the evaluation of cytotoxicity during the second year, and the last part of the third year, and the evaluation of the inflammatory markers that will finish before the end of the third year, because during the last 6 months, it will be important the communication of the data to partners and the integration of information about chemicals. And of course, during the 3-year period, partners are allowed to send us compounds to test. So the communication among partners is really essential. And that's it. Thank you.
Okay, thank you, Noemi. Questions from the board?
You.
Yes, hi. I have a question, if I may. I was wondering, what would exactly be your role in the project based on your expertise that you have acquired during your studies? What will you be exactly in charge of during the project?
Previously I have already worked with plant extracts, so I kind of have a background of about these things. But for me, it's like a new using extracts for this different and innovative techniques will be I'm very interested on it, because will be a new way to test extracts for different things that I've done before with the extract. So it's a kind of thing that I have already done, but in a new way. And also in the past, I have had the possibility, the chance to learn some techniques like the QRT PCR, reverse transcription, the isolation of the RNA, starting from different samples. So I think that there are all techniques that will be useful in this kind of project. So yes.
Okay, perfect. Thank you very much.
Sorry, a little question, if it's possible.
Yes.
How this project support safe and sustainable by design framework?
Well, I told briefly of this, but not in detail, because there were no time. Well, the European Union signed the Green Deal. There is a packet of policy I initiated with the key of create a climate neutral continent. And since a lot of substances are hazardous for the environment and the human health, the European Commission then published the chemicals and the chemical of the chemical list for sustainability that gets operationalized by the SSBD that you mentioned before. So this SSBD is like a voluntary approach that based on different aims, and one of them is, for example, bringing the innovation process towards industrial green transition. So this project is it is in this kind of context.
Okay, that's right. Thank you.
Oh, you're welcome.
Thank you so much.
Thank you.
The next one is Panesi.
Yes. Okay.
Yes.
Good afternoon, everyone. My name is Alessandro Pennesi, and I choose the project entitled Implementing Innovative Methods for Safety and problem. Okay. I choose the project entitled Implementing Innovative Methods for Safety and Sustainability Assessments of Chemicals and Materials, particularly at the nano level in the European Union. So let's start. Before beginning the presentation, I want to share with you a brief schematic representation of my short PowerPoint. So we start with the general context and the state of the art. Then we will speak about objective and experimental design, materials and methods, and finally we will see the expected results. So the CEMAT Sustain project aims to address the critical needs to improve methods for study safe and sustainable chemicals and nanomaterials within the European Union. We know that chemicals and nanomaterials today are widely used in many sectors, such as energy, housing, mobility, and like that. In particular, nanomaterials are substances with size between 1 nanometers to 100 nanometers, and this feature is very important because they have an increased surface surface as opposed to surface area to volume ratio. So this feature can differ significantly from their bulk counterparts. Even if chemicals and nanomaterials have an important role in modern life, they have also important hazardous properties for human health and the environment. In particular, in the European Union, the legislation about the use of chemicals and nanomaterials is under the control of European Chemical Agency. European Chemical Agency is an agency, so is a decentralized and organization that make regulation about the use of chemicals and nanomaterials. Then this information are sent to the REACH regulation, means regulation, evaluation, authorization of registration, evaluation, authorization of chemicals. The regulation, the REACH is a regulation of the European Commission. So in a few words, the European Chemical Agency send to the REACH which chemicals or nanomaterials can be a risk for human health and for the environment. So briefly, I think that the objectives will be the development of standardized methods for risk assessment. This means that it's possible to create protocols for a better evaluation of ecotoxicity and exposure of chemicals and nanomaterials. After that, we can have as objective an update, of course, of regulatory guidelines, and finally, we have promote sustainable practices. For the experimental design, I image three main phases. The first one is about theoretical modeling, so computational analysis. The second phase is about laboratory experimental analysis. So spectroscopy techniques, microscopy techniques, molecular interaction studies, so microfluidic systems, and omics methodologies, so transcriptomics and proteomics. The last phase is the development of in silico models using machine learning algorithms and bioinformatic software. What about materials? So for chemicals that are substance present now in the REACH regulation, I think is a good practice to start this new study with the chemicals present in the regulation. So I have listed some of them. Instead, because the legislation about nanomaterials is not full if compared with chemicals. So the nanomaterials that can be studied in this project will be considered the ones that are in literature, in scientific literature. So we consider batteries, carbon nanotubes, lithium, iron, graphene, the one that we can find in cosmetics, and for example, the one that we can find in antimicrobial textiles. About methods, so we just said that the first phase is about theoretical modeling. So the technique we can mention it here is density functional theory, DFT. That is a computational quantum mechanical modeling. The DFT finding a solution to the Schrödinger equation. So it means that this technique can finding important information for the electronical properties of the nanomaterials, and the information that came out from the DFT can be used also in laboratory experimental methodologies. For this section of experiments, I put attention on photoelectron spectroscopy. Photoelectron spectroscopy is based on the photoelectric effect. In particular, the photoelectric effect is come out when a source of light beam the electron near the surface of the materials. These electrons come out and they are called photoelectron. This phenomenon is the technique based on this phenomenon are X-ray photoelectron spectroscopy and ultraviolet photoelectron spectroscopy. In particular, the X-ray photoelectron spectroscopy consider the core electrons of the atom. Instead, the ultraviolet photoelectron spectroscopy consider the orbital electron, so the most outer electron. For this reason, XPS can be considered more in-depth endogenous technique used for study the chemical structure of the materials. Instead, the ultraviolet photoelectron spectroscopy can be used for study the electron properties of the materials. About microscopic techniques, we know that there are several techniques falling in these groups, such as atomic force microscopy, IFM, or electron microscopy, such as TEM and SEM. In particular, electron microscopy used as beam, an electron beam, so the wavelength of the electron is shorter, and then like microscope, so we can do an in-depth investigation about the composition of nanomaterials and chemicals. Microfluidic systems offer also a good scenario for this project because are dynamic system if compared with the classical with the other classical static technique. An example of a microfluidic system can be an organ on a chip that simulates for example, an organ. With microfluidic system, we can study also the toxicity, oxidative stress, and we can do also single cell studies. About the transcriptomics and the proteomics methods, we can have mass spectrometry, high pressure liquid chromatography, DNA microarray, and protein microarray. the last, how can say, the last phase of the methods can be the in silico models. So we have quantitative structure activity relationship and quantitative structure property relationship. Both consider the
I'm sorry, Alessandro. I need to remember that every student has 12 minutes.
I'm so long. Okay.
So you're, yeah, if you can mention something.
Okay. Okay, I go on. For in silico models, we have these two analysis and chemical fingerprints. And for finally, for expected results, we can have development of new methods, so computational methods, experimental methods, and in silico methods. Another important point is the identification of key parameters, such as size, shape, surface charge, and functional groups, and other type of results can be creation of new in silico models, improvement of regulatory guidelines, and integrated risk assessment and life cycle analysis methodology. Thank you.
Okay, thank you, Alessandro. Question from the board?
Yes, I have a couple of questions. First of all, thank you. It was nice to see how you presented this project related to the ChemAtSustain project. And I was wondering, why did you choose this subject? Which is the interest you had looking at this kind of project?
Thank you. So during my experimental thesis in master degrees, I was in Bologna at PhD, that is a research center where study crops, industrial crops. In particular, during that period, I studied the flavonoid pathway in cannabis with bioinformatic tools. We know that this pathway is one of is not studied a lot if compared with other secondary metabolites, and so I'm interested in this for this project because bioinformatics I think is a good solution for improving for the ChemAtSustain, and also because actually I'm working in a small company where we have a sort of microscope device for hospital. So I'm interested to the microscopy world for this reason. So the informatic for my period during my
Thesis work.
Thesis work, and thank you.
So when you see the logo ChemAtSustain,
Yes.
How do you think it is related to your project?
ChemAtSustain in the sense that chemicals, materials, and
Sustainability. Yeah.
Yes. Sorry, my English is not. I know.
No, it's fine. It's fine. You're fine. Don't worry. So chemicals and materials that you presented, all right?
Yes, and sustainability for the safe, for the assessment risk of about the use of materials and chemicals in the European Union. So.
Okay, so you appreciated this new method that has been realized in this kind of project.
Multidisciplinary project that yes, encompasses chemicals, nanomaterials, in silico investigation, experimental techniques, and some like this.
Okay, thank you so much. It's fine for me.
The next one is for Emilio Eugenio.
Good morning and good evening, everybody, and good evening to the committee. I'm Eugenio Formilio, and I participate in a PhD in Biomolecular and Health Sciences, Department of Biomolecular Sciences. The related topic is effects of Tecar therapy treatment on reducing acute and chronic post-articular and muscular inflammation and pain. The relationship between biological and functional markers and neuromuscular signaling in athletes. According to the International Olympic Committee, musculoskeletal injuries are new or recurring musculoskeletal complaints incurred during competition or training that require medical attention, regardless of the potential absence from competition or training. This recent review tells us that in most of sports examined, muscle tendon strains and ligament sprains are the most common and frequent musculoskeletal injuries. So the key question is, what is the real treatment for musculoskeletal injuries and pain? Scientific literature certifies that there are a wide variety and many combinations of effective treatments for musculoskeletal injuries and pain. For sure, in a recent year, one of most used electromedical devices for this type of problems is certainly Tecar, the capacitive and resistive energy transfer. Tecar therapies has very important biological effects on the human body, as it causes an increase in skin and tissue temperature. This is called the diathermic effect. It also determines an increase in perfusion of the skin microcirculation. And it also causes an increase in intramuscular blood flow. Precisely because of these relevant biological effects, Tecar therapy is commonly used in physiotherapeutic clinical practice and also in sports. It reduces pain in musculoskeletal disorders and musculoskeletal pathology. It's used in dermatofunctional treatments. It's important in sports injury to promote and speed up the return to play. And also, it leads to improvements in biomechanical parameters in the athletes. Despite this, there is still a lack of scientific evidence proving the effectiveness of Tecar therapy in counteracting the biological and physiological performance fatigue processes. Some of the most important biological markers of muscle damage are creatine kinase and the lactate dehydrogenase. As follows, we can see some of the relevant biological markers involved in the inflammatory processes: interleukin-1, interleukin-6, and tumor necrosis factor alpha. On the other side, as for physiological and also functional factors in performance fatigability, there are the decay in muscle fiber conduction velocity, the increase in the degree of synchronization between the firing times of motor units by CNS, and also the reduction of the recruitment threshold and the modulation of the firing frequency of the motor units. About the last points, surface electromyography is a suitable tool for studying and monitoring conduction velocity. Conduction velocity, CV, is an important parameter of functional efficiency, and it allows to understand the state of fatigue in all terms. So in health conditions and also in presence of neuromuscular disease. The signal of surface electromyography is the instantaneous algebraic sum of the electrical contribution provided by the recruited motor units in response to the activation provided by the motor neurons. Consequently, we can say that the information extracted from surface electromyography is a global measure of the activity of motor units of the dead muscle. So the aims of our project are to evaluate the effects of TECAR therapy treatment in improving conduction velocity, reducing inflammatory processes, and also resolving muscle damage following 3 different exercise protocols, which are endurance training, repeated eccentric exercise, and the counter-resistance exercise until the failure. In the first year, and anyway before the start of the experimental protocol, it's necessary to start preparing the project for ethics committee. It's necessary a collaboration with the surface electromyography expert. We also need to start a partnership with TECAR companies, to start a collaboration with physical therapists, and also we have to start working with the blood testing laboratory. In the first and also during the second year, there will be the experimental phases, the determination of the minimum number of participants through statistical power calculation in relation to the primary outcome, the definition of inclusion and exclusion criteria, the recruitment of participants, of course, the familiarization of subjects with the exercise protocols, and finally, the start of experimental protocol. In the second year, we will implement our experimental design. We will have 2 groups, a TECAR group and a control group. The difference between the TECAR group and the control group is that the subjects into the TECAR group will undergo TECAR therapy immediately post-exercise, and also 24, 48, and 72 hours after exercise protocol, while the participants into the control group will undergo the same treatment therapy, but with devices switched off. Surface electromyography and blood tests will be carried out immediately pre-exercise at the baseline, immediately post-exercise, and after 24, 48, and 72 hours exercise protocol. From the second to the third year, we will continue with the analysis of data, the discussions of results, and finally with the writing of a scientific article. Our project, results of our project could provide important indication on the effectiveness of treatment with TECAR therapy and regarding the possibility of testing innovative markers. For example, the conduction velocity extracted by surface electromyography. This is for the assessment of neuromuscular recovery. If so, this would open up new perspectives and new fronts in this field of research, such as the correlation between biological markers and neuromuscular conduction velocity. For example, the correlation between creatine kinase and neuromuscular signal conduction velocity, like the application of TECAR therapy on subjects with different types of injuries, tendonitis and muscle injuries of with different types of severity, and finally with the study of specific protocols for a more effective return to play for the athletes. For example, the number and the intensity of TECAR therapy sessions. Here are the references. And thank you all for your attention.
Okay, thank you.
Okay.
I have a question. Have you thought about the range of the age of your participants? You know, I mean, if they are young, very young, or
the participants of our project.
Yeah.
Participants will be part in our project. It's a half adult subjects with regarding the inclusion criteria will be part in our project adult subjects, and regarding
what do you mean? Sorry, what do you mean?
Adult subjects?
Yeah, the range.
Half adult subjects, so 18 year and more. The important is regarding the exclusion criteria is that the subjects are without illness, diseases, and without any injuries. And also the subject with, for example, metabolic conditions such as diabetes will not be part in our project.
I have a consequent, another question. And what about gender diversity? Because
gender
I
diversity,
didn't heard about men or women.
No, we try to choose 50-50. Of course, men and women in equal part for guaranteed the parity of in our study. Of course.
Okay. Thank you.
Thank you.
Question from the board?
I have a curiosity, if I can.
Yes, bro.
Is Tecar therapy the only treatment or the most common treatment that can be prescribed for muscular inflammation, or do you suggest other kind of treatments?
No, Prof. It's not the only, but in Italy it's commonly used because the effects and the benefits are taken for granted. But in the reality, there is a lack of studies as a scientific evidence that prove its real impact on the inflammatory processes, so interleukin-1, interleukin-6, on repairing muscle damage, and also from on the recovery from fatigue processes. So for all these reasons, we want to see all these parameters all over the time, and we see the real impact of Tecar therapy treatment in all these parameters. To answer, it's another one treatment is treatment manual treatment. So massage in combination often with Tecar therapy for resolve this type of problems. In Italy, for sure.
Yeah. Okay. Thank you very much.
Thank you, Prof. Thank you.
Oh, Di Fabrizio Ludovica.
Here I am. Can you hear me?
Yep.
Yes.
So my name is Ludovica Di Fabrizio, and I applied for the in vitro evaluation of colostrum or mature milk about the buffalo's milk. So the evaluation of the colostrum or the milk, so about the extracellular vesicles. So in my presentation, I brought a definition of extracellular vesicles. So we know that extracellular vesicles are lipid bilayer delimited particles. So they can, as we can see from the image that I put in the slide, they can be released from all the cells, almost all the types of the cells, and they can be they can origin from the plasma membrane. Can you see like the mouse or not?
Yeah.
If I move the cursor, so I'll put the thing here. Okay, so they can origin from the endosomal system, and they can even shed from the membrane. But with the term extracellular vesicles, we intend it's a global term. So we intend endosomes, exosomes, oncosomes, ectosomes that are all particles that are composed by a layer of lipidic bilayer. And in recent studies we saw, researchers saw that these vesicles are contained in a lot of biological fluids, which can be, for example, urine, saliva, or blood. But the most important thing that is about the project that I applied for is in human and bovine milk. So here there's an highlighting of the human milk. So in recent studies we saw that the presence of extracellular vesicles in human milk. We can highlight the difference with the bovine milk because in the human milk we have a higher concentration of protein that are associated with the immune and growth system. So that can be useful for the embryo to survive, for example, in case of the colostrum, or in the mature milk, the protein are always associated, this kind of protein that are present with the extracellular vesicle are associated with the immune system. To study the extracellular vesicle, we should isolate them. So here I put all the methods that I use to isolate the extracellular vesicles. The important thing in the human milk is that the centrifugation or the separation of the particles, it's easier. Easier in a way, because in the human milk, in the EVS contained in the human milk, there are less protein, less lipids, and all these other kind of particles. While in the bovine milk, here it is, in the bovine milk, so previous studies, they demonstrated that the EVS, the extracellular vesicle, they are not degraded in the environment, in the digestive environment, which is a good point because we have an harsh environment. So the degradation of the extracellular vesicle could be not so useful, because if they degrade in this kind of the environment, then maybe we can use them as carriers, because in previous studies, researchers studied the fact that these vesicles in bovine, but even in the human milk, could be used as carriers of protein, of mRNA, of molecules, so as therapeutic agents. But as I was saying before, to isolate the extracellular vesicle in a case, we're talking about the bovine milk, the centrifugation or the separation is a little bit more difficult because there's the presence of protein and lipids. So we need more passages to have a completely isolation of the extracellular vesicles. And the thing that I think is the most interesting is the fact that the extracellular vesicle remain intact, so they can carry the molecule or the hypothetic things, like I was saying, mRNA or therapeutic molecules, and can be useful in different occasions, because the extracellular vesicles has a lot of functions, and I didn't put all the function because there are so many functions, but I think that 3-4 of the best function that they observed is the gut and intestine health, first of all, that we saw before. So the fact that they don't degrade and they can be carried. The fact that there's an effect on skin and wound repairing, because extracellular vesicles have some effect on the inflammatory response. Even we have another two functions that I think I should say is the anti-cancer properties and the antioxidant properties. The anti-cancer, I found a lot of studies about this property that is maybe one of the most important, obviously. And a thing that I can add is that this is all about human and bovine. But about the project that I applied for, we are talking about buffalo milk. And as we know, the buffalo is a strong animal. And when he's an adult, he's very strong. On the contrary, the cubs are not so strong. So maybe during in farm animals, there's the problem of the widespread of the antibiotics. So we know that the resistance to antibiotics is one of the most, one of the biggest problem of nowadays. And this problem is in human, but is always in animals, even in animals. So I think that finding new vesicles, maybe studying a new composition of these vesicles in the buffalo could be interesting, because as this study says, in the buffalo milk there's a high concentration of innate immune system proteins. So I even saw that the vesicles are there's a good interaction with macrophages that we know that are part of the innate immune system. So I put some strategies that could be interesting. As you can see in the image, if you see clearly, if you can read, I hope so. There are different animals from which the extracellular vesicles are taken. So horses, cows, even goats. We are always talking about the milk. So goat's milk, cow's milk, and buffalo milks. And here there are all the function on the brain, on the immune system, on the heart, on lungs. So as I was saying, the function, the advantages of these vesicles are so many. So in these 3 years, I would like to analyze, first of all, the content of these vesicles. And we can even highlight the difference between the colostrum and the mature milk, because we saw in the bovine and in the human milk that in the colostrum there's always a higher percentage or higher concentration of protein about the immune system. And in case there are some difference between so the buffalo and the bovine milk, we can analyze the effective uptake, because one of the biggest problem of the extracellular vesicle is that we don't know the effective uptake. So we don't know if they go to the target, if they are uptaken by the target. So this could be interesting, maybe studying the different composition. Another thing that I can say it's interesting because of my previous work. During my thesis, I'm graduating, but I'm still going to the laboratory. I was able to manage a molecule that is a thiol compound, that is the I-152. And as I said before, the extracellular vesicles could be used as carriers. So if we can use them as carriers, we can use them to carry some therapeutic molecules, such as this molecule, the I-152, that is a compound that can release N-acetylcysteine and cysteamine, and this compound can modulate the redox state and can decrease the reactive oxygen species, so the ROS. So this could be interesting because I could use my thesis, my knowledge about the HPLC, all the study with the glutathione, even the highlight of the antioxidant property of this molecule, but even about the molecule of the glutathione, to study these vesicles. And the last thing that I would say is that we can observe the difference between the use of the extracellular vesicle alone, so solo, or the use of extracellular vesicle in combination with different therapeutic molecules. But first of all, I would say the I-152 for my background and all my study during my thesis. Thank you.
Oh, okay, thank you, Ludovica. Okay, question from the board.
Yeah, thank you. Yeah. Can I ask you a curiosity?
Yeah.
Did you plan also to investigate the composition of your extracellular vesicle after isolation?
Yeah. As I was saying before, maybe I wasn't so clear, but as I was saying before, the composition of the buffalo milk or the bovine milk, the animal's milk, is different from the human milk, so could be more difficult to isolate precisely the protein, or not the protein, sorry, the particles, so the vesicles that we need. So yes, one thing would be to completely isolate, because if we completely isolate, we don't have any destruction from the vesicles. So we studied that and we concentrate on that. After the centrifugation, I mean all the isolation processes, we can study the composition and have a more clear thing.
Thank you.
Thank you.
Questions?
Yes, I have one, a curiosity.
Yeah.
How do you think to plan your experimental research during the freeze?
So I would say, I don't want to repeat myself, but I would say that the first thing to do is to try the separation thing. So I think this is clear. So in the first part, I would concentrate on the isolation and separation of the extravascular.
For the first year, you mean?
Yes, I mean, it depends on the time. But in the first year, there could be the separation, okay, and the isolation, and the study of the composition that could require a lot of, I mean, not a lot of time, but could be difficult because the colostrum has to be used. Sorry for this word, maybe it's not the formal and the scientific word, but the colostrum has to be used very fast. So could be different if we have the colostrum and we analyze it in the moment, or if we have the colostrum and maybe we frozen it and we wait. So there could be some disadvantages, some barriers.
We need time to set up the system. Okay,
yeah.
This is the first year. The second one?
The second one, I would say the highlighting between, to highlight the differences between the other studies, because on the buffalo in literature, I mean, I couldn't find anything, but I don't think there are so many information. But I think that if the project can start is because the buffalo is resistant, so we can study more in the how can I say, in particularly the immune system and how to improve the immune system. Because if we improve the immune system of the buffalo, maybe the cubs won't have some problems, because in the immune system of the adult could pass to the cubs. So could be this phase, the second year could be this phase, and the highlighting of the different composition of the vesicles. The last year, I would concentrate on the difference between the other animals and the advantages, because there are advantages, even maybe on studying a new maybe the oral composition, because maybe it's more likely, we are more likely to take a pill or to an oral disponibility, and not, for example, an endovenous something. I mean, on the how to spread the vesicles, how to assume, I don't know how to say it, how to say the term in specific English, but I would go more on the therapeutic appliances, on the
applications. Okay.
Applications, sorry.
Okay.
Okay. Thank you. I see Rahman Muhammad. We skipped before. Rahman Muhammad. Okay. Good morning. Good afternoon.
Good afternoon.
Great. Okay.
I am Mohammad Rahman from Pakistan. My background is from veterinary medicine and veterinary surgery, and I have done my MPhil in veterinary parasitology from University of Agriculture, Faisalabad. Here is layout of my presentation, in which I have to discuss different points. First of all, here is my introduction. Then it is my educational background. During my MPhil research, I have selected a topic which is neglected zoonotic disease, known as hydrated cyst or echinococcus. And its prevalence was about 17% in Faisalabad, Pakistan. Here is my research topic of my research project. When I discuss about its background, extracellular cellular vesicles are lipid bilayer which are found in extrastitial fluids and in colostrum, in milk or other fluids. When we discuss about colostrum, it is the first fluid, or we can say first liquid produced after parturition or after delivery, which is rich in nutrients, protein, amino acids, lipoprotein components, and we can say less in number of sugar molecules and fat concentration. We can also say it as a yellow gold or yellow fluid or gold fluid, because it is essential for newborns, with newborn babies, either in humans or in animals, because due to colostrum, antibodies again in the body, and the immune system becomes strong. But in other cases, when we didn't feed colostrum to the newborn babies, their immune system become weak, and we can say they die in a week or in 2 weeks because of their immune system. Now here is a study gap. We can say that the immunity which induced due to colostrum, the research on that is not sufficient at all. So here is a gap which we can fill with our research. Here is a hypothesis of my research, as the extracellular vesicles which are uptaken by intestinal epithelial cells or macrophages. Macrophages, we can say these are engulfing cells. Due to these engulfing, inflammatory cells or inflammatory procedure, it becomes stopped. Here is the significance of this study is that the neonatal mortality becomes decreased when we discuss or when we start feeding colostrum to the young ones. So we can say in dairy sector, in equine stud farms, it is essential to feed the newborn minimum 3 days, and we can say colostrum from the day of birth up to 72 to 84 hours, it secretes from the mammary gland, and after that normal milk produced. Here is the methodology of my research, as we have to collect colostrum within the 24 hours after parturition or after delivery, and then we will store it at 4 degrees centigrade and preserve it for 2 hours, as the extracellular vesicles, their integrity become normal, and then isolation needed. We will centrifuge the colostrum. First, we will centrifuge it as 300 for 10 minutes to remove the cell, which will be normal cells, and then we have to remove the debris which is in the milk or in the colostrum, and we have to centrifuge at 2000 gravity per minute for 30 minutes. And for ultracentrifugation, we have to centrifuge it at 1 lakh gravitation for 70 minutes. Then we have to go for the chromatography, when we have to exclusion with respect to the size. We will use PBS for this purpose, and after the protein assay, we will see the in the fraction forms. Nanoparticle tracking analysis, and we can also say it as a NTA analyze size distribution and concentration. It will differentiate or divide into different sizes. Then we have to see it in electron microscope. We can say it as a transmission electron microscope, and we will prepare it by uranyl acetate. Then it will be its visualization will be normal, and we can say the morphology of cells. Then we will go for the SDS page or Western blotting test, and here are we have 3 markers here like CD63, CD81, and CD9, and we will use different antibiotics like penicillin, streptomycin, etc. Routine we will do quantification and as well as qualification test for it. PSA assay test we will do, which is bovine serum albumin test. All right. Then we will go for the RNA extraction. RNA extraction will be easy as we have different kits in market to isolate RNA. RNA contents we will analyze it in bioanalyzer, which is available in our labs. And I have done SDS in my previous research. I have done on Echinococcus, and I have separated it protein. Then we will label is due to fluorescent dye, as we have PKH67 dye. And then we will use chromatography for the size exclusion. Then we will do for the cell culture as intestinal epithelial cells. Definitely we have to go for intestinal epithelial cells and for macrophages for our research. Here we will use 10% fetal bovine serum and antibiotics like penicillin, streptomycin, and we will allow it to remain for 21 days. Then definitely the other procedure will go for the macrophages, and for macrophages we will use PMA, which is 4-12 micitrate 13 acetate. Then other will be the uptake assay, which is different procedures in it like incubation, flow cytometry, microscopy, immunomodulatory activities, etc. Here are different procedures like we will go for the different concentration and for different time periods like 10 microgram, 50 microgram, 100 microgram, and for 1, 4, and 24 hours different time periods. And then harvest it and wash with the PBS solution. Then we will go for the flow cytometry, and we can use fluorescence intensity using the flow cytometry. Then microscopy fill cell in 4% formaldehyde solution, stain cell nuclei with DAPA. Immunomodulatory activities in which we will use different enzymes for this procedure. Then for the likewise for the macrophages, here we use LPS 100 nanogram per milliliter. Then we will go for the gene expression analysis. After RNA extraction, we will go for the gene expression. As from gene expression, we can easily manage our cells. We will collect the supernatant from our treated sample, and we will go for the ELISA. Here are different procedures from which we can see its quantitative analysis, qualitative analysis, its phenotypic analysis. and the results will be obviously good after the procedure. And likewise, when we discuss the result after our procedure, we can easily say which will be helpful in market as the efficacy and efficiency of colostrum feeding. Here was my small PPT. Due to time issues and due to other technical issues, some part of my PPT I can't show that.
Okay. So
once again, sorry for interruption, ma'am, due to electricity and due to some other technical
yeah,
issues.
no worries, no worries. We know these things can happen. Don't worry. I have a question, if I may.
Sure, ma'am.
From what I see in your CV, you have been studying zoonotic diseases. So your expertise seems to be a little bit far from this topic area that you've chosen. So I was wondering, based on your expertise, now, what would you be your contribution in the project? Why have you chosen this specific topic for your PhD application?
As ma'am, we can say in animal sciences, likewise in the dairy sector or equine sector, colostrum feeding is one of the essential needs. And in zoonotic diseases, we can say mortality issues, and due to colostrum feeding and due to these research, we can manage these issues in animals. We can improve their immune system, which is the major issue nowadays. Their immune system obviously increased due to colostrum feeding.
Okay, so you plan on studying the effect of these vesicles on the animals, not on the human host, for example? Or are you planning on doing some microbiological analysis on the colostrum as well?
Yes, ma'am. Yes, ma'am.
Well, both or either one? I'm sorry. I'm sorry. Could you please repeat your answer, because I haven't heard it.
There is an interruption.
Yeah. Could you please repeat your answer? Because I didn't hear it.
Yes, ma'am. I will consider both studies, as colostrum feeding is essential for both, obviously for animals, for humans.
Okay, and what about the microbiological part?
Yes, ma'am. In case of biological, we can say as one health sector in which human, animal, and environment collaborate with each other.
Okay, well, I think I've got what I needed, I think. Okay.
Saki-san.
Hello, Professor. Okay, hello everyone, and a good day to you all. If it's all right with you, I would like to share a bit of background about myself before we begin. My research project title is In Vitro Evaluation of Colostrum and Milk Extracellular Vesicles Uptake and their Immunomodulatory Activity on Intestinal and Macrophagic Cells. My name is Sahar Saki. I'm from Iran, living in Karaj. I received my bachelor's degree in microbiology. Then I completed my master's degree in genetics, and thesis is about production of nanocapsules containing immunotoxin consisting of diphtheria and herceptin, and evaluation its function on breast cancer cell line. During my work experience time in one of the company in Iran, I worked extensively on the cloning and expression of various recombinant protein in prokaryote hosts such as E. coli. I'm skilled in cloning, protein expression, protein purification, cell culture, Western blot, and preparation of nanoparticles such as chitosan. And one of my key achievements during my academic career was publish article and go to international conferences and speak these some of international conferences and courses such as molecular medicine and nanovesicles. And over the past 2 years, my focus has been on the production of recombinant proteins and drug delivery. These experiences and my keen interest in the field of drug delivery led me to the project I aim to undertake in vitro evaluation of cholesterol and milk-derived extracellular vesicles and their immunomodulatory activity on intestinal and macrophagic cells. The objectives of these projects are to investigate how these extracellular vesicles to taken up by intestinal and macrophagic cells and evaluate their immunomodulatory effects on immune response. The finding from this research has the potential to development therapeutic strategies for gastrointestinal to develop new therapeutic strategies for gastrointestinal disorder and improve probiotic formulation. The first stage is extraction of extracellular vesicles from cholesterol and milk using centrifugation and filtration. Then be purified and characterized using microscopy and analysis. Next is cell preparation. We will culture intestinal and macrophage cells in appropriate media in ensuring the right conditions for cell growth, and maintenance is crucial for the success of subsequent experiments. The evaluation of extracellular vesicles uptake in this stage, extracellular vesicles will be labeled with fluorescent dyes and added to the cell culture media. Their uptake by the cells will be assessed using fluorescence microscopy and flow cytometry. Next, assessment of immunomodulatory activity. We will evaluate the immunomodulatory activity of the extracellular vesicles by analyzing that expression of immune-related genes using RT-PCR, measuring cytokine production with ELISA test, and examining change in cellular activity through immunocytochemistry assays. And finally, data analysis and interpretation. This project is important because it aims to provide a better understanding of the role of colostrum and milk derived of extracellular vesicles in regulating the immune system. It advances scientific knowledge and intercellular communication and immunomodulatory mechanisms. And the potential application of this research include the development of new therapeutic strategies for inflammatory and gastrointestinal disorder, as well as the improvement of therapeutic formulation to support gut health. And my interest in the drug delivery field and my past experience in genetics, recombinant protein drug delivery with nanoparticles align perfectly with the purpose research project. I am excited about the opportunity to work on this project and expand my research in this advanced field. Thank you for your attention.
Okay, thank you.
You're welcome.
Some questions from the board? No?
Hi.
Okay.
Go ahead. No, go ahead.
Okay. Starting from your experience or studies preparing this presentation on this topic, of course, do you expect that EV in colostrum and milk are the same?
Please explain more that. I can't hear you, unfortunately. I'm so sorry. Do you hear me?
No, I think colostrum and milk are two different types.
Yes.
Of
yes.
Period.
Yes.
Okay. So, do you expect that EV are the same?
I think it's two different parts.
Simple question.
Yes, but I think it's different.
Manuela, go ahead.
Yes, you said at some stage, you said that your background in genetics will help clarify some issues in the project. Can you elaborate on this part a little bit more, please?
Yes. In my master's time, I researched about nanoparticles such as chitosan and drug delivery with about cytotoxicity in breast cancer cell line such as MCF7. And I studied about cloning and expression recombinant protein such as diphtheria, Herceptin immunotoxin, and I studied about cytotoxicity in breast cancer cell. And I like to this drug delivery topics, and I like to this research project.
Yes, but in this research project, the aim is not to encapsulate anything the extracellular vesicles, is to see whether there are immunomodulatory activity of the vesicles. So this is something new that you haven't done. Am I right?
Yes, it's new for me. and I like this topic. Justin?
No, I was just wondering whether your expertise would bring any benefit to the project. Just a curiosity.
Yes. I think it's my expertise in recombinant protein expression and nanoparticle techniques, and my committed to laboratory work and well prepared me for contributing to ongoing research and more research about these extracellular vesicles.
Okay, thank you very much.
You're welcome.
Maybe depends. Kadzeni Mohammed, it's on my list, I think.
Okay.
Yeah.
Yes.
So,
yes, Professor. Good evening.
Okay.
Good evening.
Good evening.
The clozapine inhibit the adipogenesis and mitochondrial biogenesis, differentiating adipocytes. So in this research, we are going to an identification of the mechanisms and development of the pharmacological strategies for their prevention. So before start to give my hypothesis, I would like to, give you a general overview, because for all of us, I think it's somehow unknown and ambiguous area. So I think I have to talk a little more about the general overview. The prevalence of metabolic disorders has been increasing worldwide, partly attributed to side effects of certain pharmaceuticals. Clozapine, which the drug we are using in our research, is a second-generation antipsychotic drug, and it's so effective in treating the very, strict schizophrenia. But unfortunately, it's associated with some negative side effects, like the inhibit the adipogenesis and the effect on the insulin resistance. The exact mechanism underlying these effects remain poorly understood, but I'm going to show you the recent studies about this drug, which this drug can inhibit this enzyme NADPH oxidase, and also some contracts in cytosolic area in H2O2. And also this drug, as I said, that impaired the mitochondrial function. So let's see about some characteristic and feature of this drug. Is, as I said, a second generation antipsychotic and treat the schizophrenia, and it has a lipophilic nature, and its response is to inhibit the activity of several enzymes, induction of impairment of mitochondria, and inhibition of NADPH oxidase 2, and inhibition of nuclear factor NRF2, which this nuclear factor stimulates the pathways that result in inflammation by reducing oxidative stress, inhibiting pro-inflammatory signaling, and restoring the function of mitochondria. Okay, as a schematic summary of the function of this drug, I took it from the paper recently published by the collaboration between the Urbino University and one university in Belgium and another university in Hungary. It's very fascinating in this area, because, as I said, that it's a very unknown area and it has a lot gap to do their research. Okay, by the use of these specific cells, SW872 cells, which is related, it's cancer cells related to fat cells. Here we don't use this drug, clozapine, but here we use it, as we can see that it inhibits the this enzyme, NADPH oxidase 2. So as a result and output, we will have the less NRF activation, it's the transcription factor, and less adipogenetic signaling. And in a later slide, I will show you the results which belong to investigation of some biomarkers, NRF2, PPAR gamma, and CEBP beta. So it's a schematic and summary overview of the function of this drug. What we are going to in the future, we have the three areas in order to research. Firstly, try to understand the mechanisms of some things. For example, try to know about the pathways when we use this drug, and try to detect and discover new biomarkers in order to publish the new paper. Just we can, for example, focus on those biomarkers by biology methods, can assess them and publish the paper. And the most hardest part is to go through the pharmacological modulation. So just we have these three areas to go on in future. Before to show my hypothesis about this area, in order to know, this is from these results from the recent article which I told you from the collaboration of Urbino and other universities. We wanted to know where we are staying now and what we are going. It helps a lot to what we are going in the future. It's from the result part. So the function of clozapine can, decrease the adipogenesis. So here we have two biomarker in order to reach to this conclusion. So CEBP beta is a transcription factor. If we have less of it, it means that we have a problem in adipogenesis. So we have a less adipogenesis. The green column is related to, and also these are from the obtained from the cancer cells. This T0, it's for the beginning of the research and examination. T3, it means that after 3 days. And this one, the yellow column, it's belong to the control one, and the blue column, it's belong to when we use this drug clozapine. So as we wanted to see, we have a less amount of the in the blue column, so we can see this and conclude that this drug has this negative effect. Also, another result it's that the clozapine suppress the reactive oxygen species and also the activation of NADPH oxidase 2. And also here we have two other biomarkers, P47 phosphorylation and P67 phosphorylation. For both of them, but this one it's a little, but this one is higher. We have the less amount of the when we use the clozapine, so we can conclude that the clozapine inhibits the activation of this enzyme. And another result also it has a negative impact on this transcription factor NRF2. It's make the less expression. So we can see that also these are obtained by the PCR method, and these are obtained by the Western blotting. And also this one, it's related to the impairment of mitochondrial function. So it shows the mitochondrial reactive oxygen species indicator. Here we can see that if we have the more amount of these mitochondrial reactive oxygen species, we can conclude that okay, we have a problem in function of our mitochondria. And so in terms of the pharmacology, as all of us know that it's a so difficult part and so time consuming. So the hypothesis would be that develop and modify, this drug in order to, make it more, antioxidants. The feature make it more, and the develop the small molecule to, mitigate the inhibition of NADPH oxidase 2, and also the modulate the this transcription factor NRF2 or other transcription factor to induce the activation. Maybe my hypothesis is that maybe by change of, because it's so lipophilic, this drug, maybe by change of the amount of and of the lipophilic feature, or by the change of the polarity. It's a very accessible, but if we are going to add some molecule or add some add some atoms and make the change the structure of the molecule, so we have to use another tools, and we should take help from the bioinformatic tools for artificial intelligence, because so it's a it's a drug, and it takes a lot of time to modify and even discover the new drug. It's a very difficult part. But in terms of the understand and investigate mechanisms, so we can consider as a mechanical understanding. We have a PGC1 alpha. It's a master regulator of mitochondria, and it's the activator of the energy metabolism. We can actually study about this, and its hypothesis is that I think this clozapine interfere with the signaling pathway, which this signaling pathway activates this regulator, and so we can work on this and show it to other people who publish the paper. Okay, it's the mechanism of this regulator, and also the easiest part is to discover or detect the other biomarkers. For example, as I showed that we had several biomarkers, but here also we can examine the TNF alpha, interleukin 6, and interleukin 1 beta. They are for inflammatory cytokines, and our expected result is that when we assess these cytokines, we have a less after we use the clozapine. Also, we can assess the amount of ATP production is related to mitochondria, and also another transcription factor NF-kB. We can assess that. Thank you a lot for your attention. Sorry, I think I over the 20 minutes. It exceeded.
It was 12 minutes.
12, yeah.
Fine.
I can start with a question, maybe.
Okay. How do you plan to organize your research during the 3 years of the PhD?
You mean the time schedule of the?
Yes, during the 3 years.
Yes, but as I said that in for example in bio detect the biomarker and investigate about the mechanism. I think, for example, each of them maximum takes 1 year. But if we wanted to go on to pharmacological part, for example, try to optimize our drug, I think it takes at least 2 years, because we had to also, if we take positive results in vitro, we have to examine it in the animal models, and I think it's so time consuming. It depends on the point of view of the supervisor to does she or he agree with this. But about the biomarker investigation and mechanism, I think each of them takes 1 year, I think.
So for the first year, you think that you are going to be concentrated on the mechanism and then on the effects in vivo, probably depending on the systems that you can have available.
Yes.
Thank you.
Thanks from all of you to be patient.
Other questions?
Yes, I have a question. It's not exactly my field of expertise, but I was wondering whether you are planning on performing some screening to find new molecules that might inhibit the pathway that you're looking at, and if so, how would you plan this type of screening?
I just detected, as I mentioned, for example, this one in the mitochondria, but I have to study about the screening, as you said, that the parts. But just I selected, okay, this one we can study on it about the mechanism. But as you mentioned, that I didn't investigate and inspect about the screening. I should study more.
Okay, because you said you wanted to modify an inhibitor that is already available, right?
I told you about the drug, the modified. In terms of the pharmacology, it's a very time consuming.
Yes, exactly.
As I said that, we can, for example, modify in a very easy term. For example, it's so lipophilic drug. We can, for example, decrease the amount of lipophilic and see the results, or modulate the polarity of the drug and see the result. It takes a very less time compared to the let's bind the new atoms to it and see the result. So in this case, I think we have to use the deep learning, artificial intelligence, and these bioinformatic tools, because we don't have we just have 3 years. We don't have it.
I agree with that. Okay.
20
thank
years.
you. Of course. Thank you very much.
Thanks from all of you.
A question? Do you have expertise in bioinformatics for seeing molecules on some tools?
I'm not a proficient. I'm to some extent, I would say that, because my bachelor degree was completely related to engineering, and I learned the MATLAB and SPSS, and I'm proficient at them. But now at this time, I'm trying to develop myself in Python. I think I'm in the intermediate level, and also I'm going to the omics tools. I'm going to learn those tools. But yeah, generally I made a plan to maybe for next year or 2 years later I have to be proficient in the bioinformatic tools. Yeah, but now I'm in basic. I'm learning.
Okay, thank you.
Thanks from you.
This time, the last one is Cocchia Lorenzo.
Yes.
Yes.
Okay, I start to speak about my research proposal on the priority team project number 4 on the clozapine. As we know, the clozapine is an atypical second-generation antipsychotic used in the treatment-resistant schizophrenia or bipolarism and other psychiatric disease. This drug presents several advantages. For example, it has extremely effective in the treatment of defined positive symptoms and resistant schizophrenia. And then, as the second-generation antipsychotic, present fewer extrapyramidal side effects. But in the same time, it has several disadvantages. For example, it has numerous side effects like the weight gain, insulin resistance, dyslipidemia, hypertension that can lead to the onset of metabolic syndrome. In this onset, we can see, and we have several papers in the literature that say that there is a potential role for the mitochondria in the onset of this syndrome treated with the clozapine. In previous papers, I spoke about the differentiation process of adipogenic cells, in this case the SV872 cells, from pre-adipocyte to mature adipocyte, and we can focus on two different phases. The early phase with the formation of NOx2 dependent cytosolic ROS, and the late phase with formation of mitochondrial ROS that can lead to a mitochondrial dysfunction. Adding the clozapine in the differentiation medium can slow the adipogenesis, can reduce the formation of NOx2 dependent cytosolic ROS with a double effect, because the clozapine has a scavenger effect for the cytosolic ROS and then inhibit the NOx2. This can prevent the activation of NRF2 dependent antioxidant response and can anticipate the formation of mitochondrial ROS, and this situation can lead to a mitochondrial dysfunction. So in this project, there are two aims. First is to understand the mechanism by which the clozapine affects the mitochondrial function and the later events that lead to the metabolic dysfunction associated with the metabolic impairments, and then to focus on the possible intervention to counteract the clozapine-induced mitochondrial dysfunction with different approaches, with the aim of preventing mitochondrial damage or increasing the mitochondrial biogenesis. We can use several methods. For example, to evaluate the effect of the clozapine on mitochondrial biogenesis, we can evaluate the expression of mitochondrial biogenesis key transcription factor, like, for example, PGG1 alpha, and various mitochondrial proteins, for example, of the respiratory chain complexes, through the PCR real time and the Western blot assays. Then we can evaluate the quantification of mitochondrial DNA by, for example, the qPCR, and then the determination of mitochondrial mass through the use of mitotracker green or the mitochondrial ROS production with the mitotracker red by cytophotometric assays. Then we can evaluate for the first aim the effect of the clozapine mitochondrial function and the redox state. For example, we can evaluate the redox balance with the thioredoxin and glutaredoxin system activity that will be evaluated in presence of increasing concentration, for example, from 1 to 15 micromolar of clozapine. And then we can evaluate the OXPHOS capacity. For example, the oxygen consumption rate will be determined under the various conditions by the Ouroboros resolution respirometry in the first models. And then we can evaluate the bioenergetic efficiency. For example, we can evaluate the ATP levels through the HPLC-assay, and then the lactate formation, for example, with the spectrophotometer. And then that will be determined in cells incubated for short periods, for example, from 1 to 4 hours, with rotenone, that is a complex 1 inhibitor, and the 2-deoxyglucose, that is a glycolysis inhibitor, to determine the relative mitochondrial versus the glycolytic efficiency for ATP synthesis. Then we can evaluate the mitochondrial accumulation of the clozapine and its metabolites. For example, you can test if the rapid clozapine metabolism accounts for the lack of antioxidant effect in mitochondria. And then we can evaluate the mitochondrial toxicity of the metabolites. For example, through the oxygen consumption, the ATP production, and the viability, and in the differentiation cell. And then these assays, for example, for the metabolites, can be evaluated through the mass spectrometry assay. And the second aim is evaluate the new therapeutic approach. For example, we can evaluate the potential beneficial effect of various agents that specifically target the mitochondrial ROS, the so-called mitochondria targeted antioxidant that How Jiang et al. Exposure, for example, could be mito-Q, a mito-E, or for example, the mito-TIMPO. Then we can evaluate the therapeutic approach, the evaluation of the potential beneficial effect of various agents, specifically increasing the mitochondrial biogenesis. For example, the metformin, that we can see that the metformin upregulate the MPK and then the PGG1 alpha, that is an important marker for the mitochondrial biogenesis. For the project plan, what we expect, so the expected results, in the first year we can evaluate the clozapine effect of the mitochondrial biogenesis, and we expect to identify the mechanism whereby the clozapine inhibits the mitochondrial biogenesis that is so relevant for the physiopathology of the adipose tissue. In the second year, we can move the evaluation of mechanism whereby the clozapine induces the mitochondrial dysfunction, and we can expect several results. For example, we can expect to determine the impact of impaired antioxidant defense of the mitochondrial dysfunction and to observe the prompt mitochondrial metabolism of the drug, leading to the formation of the metabolites. And then we can we could expect to observe an increased activity in the residual mitochondria due to sustain the ATP synthesis required for the lipogenic process, for example. And this enhancing capacity is most likely energetically non-efficient due to the concomitant ROS formation. And in the last year, we can evaluate new therapeutic approach counteracting the clozapine side effect associated with metabolic syndrome. And we can expect to obtain results leading to the identification of drugs ameliorating the mitochondrial dysfunction and enhancing the mitochondrial biogenesis. So we expect to select the potential candidates for the future clinical studies testing their effectiveness in counteracting the metabolic syndrome in patients receiving the treatment with the clozapine.
Okay, thank you, Lorenzo. I have a question for you.
Yeah.
I have seen that you are planning to make some essays like qPCR, correct?
Yes, yes.
I'm wondering why you didn't plan to make some sequencing of the genome of metals to see if there are mutations in specific genes, for example.
Mainly by the qPCR, we can evaluate, for example, the gene expression, for example, for the differentiation, or for example, in factors as the PGC1 alpha, as I said. So the qPCR could be interesting, could be useful for that.
Okay. Thank you, everybody. Have a nice.
Thank you very much. Thank you.
Thank you, professors.
Okay.
Have a nice day.
Thank you. Bye-bye.
Thank you. Bye.